So our main question is, does the temperature and or season of a prescribed fire influence the success of spotted knapweed? And to answer this question, we had to ask a couple more focus questions, such as, how do different temperatures of fire influence catechin in the soil? Which treatments are most effective at removing spotted knapweed? And which planted plants survive best under which treatments? So to start off, we set up 61 square meter plots. They're located just a short walk away in an area of the prairie that has unfortunately been infested with spotted knapweed. We divide these 60 plots into 3 different treatments. We have 20 control plots, which don't experience any fire throughout the season, 20 low temperature fire plots, and 20 high temperature fire plots. These plots were then evenly divided between spring and summer treatments. So that means half of them are sampled and burned in the spring, half of them are sampled and burned in the summer. After setting up plots, we moved on to vegetation sampling. Vegetation sampling allows us to observe and compare any changes in plots in response to our different treatments. So we start by recording all species that we observe in each of our plots. From there, we move on to our cover estimates, which we do by, using the point intercept method, which involves taking a pin and dropping it 54 times in a grid in each plot, and then recording each species that that pin touches after each drop. Finally, we move on to biomass harvest, which involves taking a 10 centimeter strip of every plot, harvesting, drying, and weighing all the plants that we find in that 10 centimeter strip. I should note that vegetation sampling takes place before and after each burn for purpose of comparison. And then we move on to the fun part, the burning. We use a propane torch to burn each plot, and that involves holding the torch over the plot for either 5 or 15 seconds. The 5-second intervals allow us to reach our low temperature of 103 degrees Celsius, and the 15-second interval allows us to reach 316 degrees Celsius. That's our high temperature. We chose these temperatures because they fall within the typical range of a prescribed grassland fire. And here's a nice action shot from our spring burn that took place this last May.
Okay, so after we burned, we planted native plants that are typical to a Michigan prairie ecosystem. And we did this to act as a bioassay to determine if our treatments were effective. So we would expect that if a treatment was effective at reducing spotted knapweed or reducing the amount of catechin in the soil, that the plants would have a higher growth rate or a higher rate of survival. So we planted 3 forbs, lupin, butterfly milkweed, and thimbleweed, and 3 grasses, Indian grass, switchgrass, and little bluestem. We planted half of our plots with seeds and the other half with plugs, and then if rainfall was below average for the week, we would water them. And then one thing to note is that we had really high rates of herbivory with our lupin and butterfly milkweed plugs. So we're thinking that planting plugs the first year after burning may not be the best option. Here is Zach and I planting after our first spring burn. And then this is something that we found in our plots, which could potentially explain some of the herbivory that took place. Another aspect of our project is catechin analysis. So before burning and after burning, we collect soil samples, and we're going to take these back to the GVSU Chem Lab to do HPLC analysis, and this allows us to extract the catechin from the soil and directly measure how much catechin is in the soil. And the idea here is that the catechin levels will vary between treatments. We've somewhat successfully been able to do this earlier, but we're still working on a really good method to extract the catechin. So far we have results from our vegetation sampling before the spring burn, and we found 25 total species, 14 non-native species, and 11 native species. From our point intercept vegetation sampling, we found that spotted knapweed has 24% cover in our plots. From our biomass sampling, we found that 88% of the species were non-native, 12% were native, and then actually 31% came from spotted knapweed. And then these results are going to be compared to the vegetation sampling that we do later this summer, as well as the vegetation sampling that comes from the summer burn, which we just conducted today. So, like I said, we're going to replicate these methods for the summer burn. So I'll be planting later this week or next week and doing more vegetation sampling. We're going to compare data between treatments and then analyzing soil cadmium. We're hypothesizing that the high temperature treatments will be most effective at reducing spotted knapweed and helping native species establishment. We also hypothesize that spring burns will be most effective because a lot of the literature that we've read has suggested that. And we also hypothesize that summer burns will reduce the most catechins simply because summer is the prime growing season for spotted knapweed, so there will probably be more catechin in the soil during the summer. Does anyone have any questions?